let's continue our conversation about the types of non-coding RNAs. So remember that red represents the long non-coding RNAs, blue represents the short regulatory RNAs, the green are variable, they can be either, and black is neither. So rRNA makes up the ribosomal subunits, and we know that there's a 5S, a 18S, a 45S, so they come in different lengths. Their job is to bind together with proteins to create the ribosomes that function in translation. We have antisense RNAs. Antisense RNAs tend to be complementary to certain mRNA sequences, and they can bind to them and stop them from being translated. Now, this idea about microRNAs and small interfering RNAs, they both function in a process called RNA interference. In RNA interference, these two types of RNAs, their job is to go bind to a sequence that they're complementary to and inhibit translation or cause cleavage and degradation. So some differences between the two. They are about the same size. Micros are endogenous, meaning they are coded within our genome, whereas small interferent are exogenous, and they usually come from viral genomes. MicroRNAs function in gene expression, normally during embryonic development in plants and animals. They regulate over 60% of our mRNAs, and there's about over 2,000 genes that code for microRNAs. Small interferon, their job usually is to help fight viral infections, and they have also been used experimentally by scientists. So this is how they function. This is RNA interference. So if we start at the top and we're looking at how we would create a microRNA. So we start with the pre, it has to be processed to become mature. But this pre-RNA here and this pre-siRNA, and this again, viral or experiment, is going to be cleaved so that it's about 20 to 25, and this is through dicer. Now, we're going to get rid of this double-stranded through the RIS complex, and now we're going to have one strand serve as a template. It's going to go to a region that is complementary to, bind to it, and cause the degradation or bind to it to stop it from being, well, eventually it will be degraded. We'll leave it with degradation. So you're transcribed as a longer molecule, processed to be mature through dicer and joining part of RISC. Complementary base pairing cause degradation. This is RNA interference. We have small cytoplasmic RNAs. Their job is to help get our proteins that are created from where they're created to where they need to be to function. And we do this by a tagging system. So we put a sequence on the RNAs, and then we have proteins that come along and take them where they need to be. So one is this RNA component of the signal recognition particle. So we see here's an ER signal sequence, meaning this needs to go to the ER. So as it's being created, we have this signal recognition particle that binds to it, and it's going to allow it entrance into where it needs to be. So think of this as a tagging system, because remember that we make proteins at the ribosomes, usually in the ER, but they can be in the cytoplasm depending on what type of protein they are. And then they need to get where they need to be. So if it's a nuclear protein, we see here it needs to get back into the nucleus. If it remains in the cytoplasm, it has a certain thing. If it's exported out of the cell. So depending on where it needs to go, we need to be able to get it there. Then we have viral RNA. So the genome in a virus can be double or single-stranded RNA. And now CRISPR. So CRISPR is a very interesting concept. So think of CRISPR as being the immune system of a bacteria. And so this CRISPR-Cas9 is the defense. Okay, so what happens is we get in our bacteriophage. And now we have our CRISPR processing, and then we're able to recognize it when it comes again. So here we have our CRISPR. And this is our Cas9 genes that it's creating. And then we have this mechanism that has been simplified where we have this bacteriophage that infects our bacteria. And now our genes are expressed, and those proteins form a complex with it that's going to cleave it into smaller pieces. After it cleaves it, it inserts it into the genome, okay? So remember, we had our original genes here. Now we have inserted a bacteriophage. After we have it, we create these CRISPR RNAs, and these CRISPR RNAs are complementary to these sequences. So now, if it encounters it again, it's able to go and cause cleavage. Okay? So this is like a memory system saying, hey, I've seen you before. And this is only in bacteria. But we have found a way, so humans, researchers have found a way to use this system to our advantage to be able to insert genes or change sequences to solve genetic disorders. We have our Piwi-interacting RNAs. So we remember transposable elements and how they can come out of one and insert themselves in another, depending on if they are simple or retro. Well, we have Piwi-interacting RNAs that silence these elements in animals. So they are able, through a process where we have the pre-molecule and we're going to cleave it, and we get these Piwi RNAs, and then they interact with the complex that allows them to then go where they are complementary to this transposable element RNA and cleave it. And you'll start to see a pattern here where there's a lot of cleavage that goes on when we are interfering or inhibiting processes. And the last one we'll talk about is guide RNAs. So we talked about this briefly in the previous lecture where we said that during transcription that one way we can modify our RNAs is these guide RNAs. So we have a DNA sequence, we create the mRNA. Well, now we have a guide RNA that's kind of similar but not identical to an mRNA sequence. It can align with it and now use itself as a template to edit the RNA sequence. And it can do this by adding base pairs, taking out base pairs, or modifying those that are there. Okay? So now this is the second half of our non-coding elements. So we've talked about 15 of them. Understand the basics of, are they long or short, which organisms do they function in, and what is their job?